is. So let me try to explain it like I explain everything, with snack metaphors and questionable logic. You got DNA, right? Little long strings of genetic spaghetti. PCR is like making a whole bunch of copies of one specific noodle so you can study it, like making a million copies of your favorite bacon strip. Why would you do that? To find out if a certain gene is there, or to see whose DNA was left on the cookie jar mysteries? First, you need the recipe, the DNA you want to copy. 2 short pieces of DNA called primers that stick to the start and end of your target, free nucleotides, ATCG, like building blocks, and a special heat-loving enzyme called Taq polymerase that can work after you boil stuff because science loves drama. The machine doing the work is a thermocycler. It changes the temperature over and over. Why the heat? Well, you heat to separate the 2 strands like unzipping a jacket, then cool, so primers can stick, then warm a bit so Taq can add matching nucleotides and make new strands. That cycle repeats 20-40 times, doubling copies each round, and soon you've got millions. Sounds simple, but what could go wrong? Contamination can make you think you have target DNA when you don't. Choice of primers matters a lot. Do they match your target, or do they stick somewhere else and make junk? And do we always want more copies, or should we worry about privacy, ethics, or how this tech might be misused? Real-time PCR lets you watch amplification as it happens, so you can measure amounts, not just presence. Isn't it wild that a few ingredients and temperature changes let us detect tiny traces of DNA and solve crimes, diagnose diseases, and even study ancient stuff? So who's up for bacon now?